Reflex action. A reflex action, also known as reflex, is an involuntary and nearly instantaneous movement in response to a stimulus. When a person accidentally touches a hot object, they automatically jerk their hand away without thinking. A reflex does not require any thought input. Example: knee jerk, withdrawal of hand from hot objects, etc. The knee jerk is a reflex action. Then reflex arc. The path taken by the nerve impulses in a reflex is called a reflex arc. The reflex arc is a typical type of neural circuit that begins with a sensory neuron at a receptor, a pain receptor in the fingertips, and ends with a motor neuron at an effector, skeletal muscle. This travels to the spinal cord where it passes by means of a synapse to a connecting neuron called the relay neuron situated in the spinal cord. And sensory cells carry input from the receptor afferent impulses to a central interneuron relay neuron which makes contact with a motor neuron. The motor neuron carries efferent impulses to the effector muscle which produces the response. Or most reflex arc usually involve three neurons. The stimulates the pain receptors of the skin, which initiates an impulse in the sensory neuron. This travels to the spinal cord where it passes by means of a synapse to a connecting neuron called the relay neuron situated in the spinal cord. The relay neuron in turn makes a synapse with one or more motor neurons that transmit the impulse to the muscles. Reflex of these types are usually involved in maintaining homeostasis. Examples of simple reflex: contraction of a muscle in response to stretching, a blink of an eye when the cornea is touched, the salivation at the sight of food. The reflex action: involuntary response to a stimulus, brain, spinal cord, relay neuron, reflex arc, sensory neuron, motor neuron. Afferent neuron acts on sensory, spinal cord, efferent neuron acts on motor, muscle, effector, action, finger, receiver, source of heat. The classification of reflex arc. Reflex arc is classified on the basis of development, innate reflex and acquired reflex. Innate and acquired reflex. Response: autonomic reflex and somatic reflex. Complexity: monosynaptic reflex and polysynaptic reflex. Then processing site: spinal reflex, cranial reflex. Innate and acquired reflexes. Innate reflex: connections formed between neurons, genetically or developmentally programmed, generally appeared in predictable sequence. Then example: simplest withdrawal to complex subtly. Innate reflex. Then acquired reflex: learned rather than pre-established, enhanced by repetition. Then autonomic and somatic reflexes. There are two types of reflex arcs on the basis of response: autonomic reflex arc affecting inner organs, somatic reflex arc affecting muscles. The main difference between the somatic and autonomic systems is in what target tissues are effectors. Somatic responses are solely based on skeletal muscle contraction. The autonomic system, however, targets cardiac and smooth muscles as well as glandular tissue. Monosynaptic and polysynaptic reflexes. Monosynaptic reflex: when a reflex arc consists of only two neurons, one sensory neuron, one motor neuron. It is defined as monosynaptic, no interneuron. The monosynaptic stretch reflex, or sometimes also referred to as the muscle stretch reflex, deep tendon reflex, is a reflex arc that provides direct communication between sensory and motor neurons innervating the muscle. Which one? Muscle tendon reflex, muscle stretch reflex, deep tendon reflex. In the case of peripheral muscle reflex, patellar reflex, brief stimulation to the muscle spindle results in the contraction of the agonist or effector muscle. Faster response. Steps of patellar reflex: arrival of stimulus and activation of receptor. Arrival of stimulus and activation of receptor. Patellar tendon tapped by physician. Quadriceps tendon receptor stretch. Activation of sensory neuron. Receptor stimulates sensory neuron that extends into spinal cord. Sensory neuron synapses with motor neuron. Information processing in CNS. Information processing in motor neurons in body. Activation of motor neuron activated and action potential is generated and propagated. The response of peripheral receptor. Stimulation of skeletal muscle fiber leads to contraction of knee extensors. Activation of motor motor neuron, then response of peripheral receptor. Step 1, activation, arrival of the stimulus and the activation of the receptor stretch. Then activation of a sensory neuron, information processing in the CNS, activation of a motor neuron, response of a peripheral factor, then contraction response effector receptor, the patellar reflex, a stretch reflex, and the best known monosynaptic reflex. Polysynaptic reflex. Most complicated than monosynaptic reflex involves interneurons. Interneuron control more than one muscle groups. Withdrawal or flexor reflex is an example. Withdrawal reflex. Move body parts away from stimulus, pain or pressure. Flexor reflex, pull hand away from the hot stove. Hot object, pain receptors in skin, direction of impulse, dendrite of afferent neuron, muscle contraction and withdrawals being stimulated, axon of afferent neuron, cell body of afferent neuron, axon of afferent neuron, cell body of interneuron, cell body of afferent neuron, and spinal cord. Spinal and radial reflexes. Spinal reflexes occurs in nuclei of spinal cord, two types, single segmental within the one spinal segment, intersegmental, multiple segmental. Radial reflexes occur in nuclei of brain. Then Golgi tendon organ and inverse stretch reflex. Stretch receptors located in the tendons and musculoneurotic junction. It is stimulated when the tension is produced in group of muscle fibers to which the organ is connected. It detects the muscle tension in each small segment of muscle. 10 to 15 muscle fibers are connected in series with one Golgi tendon organ. Each Golgi tendon organ basically consists of a group of nerve endings covered by a capsule of connective tissue. Muscle Golgi tendon organ, nerve fiber, tendon. Golgi tendon reflex, also called the inverse stretch reflex, autogenic inhibition, tendon reflex. It is an inhibitory effect on the muscle resulting from the muscle tension stimulating Golgi tendon organs of the muscle, and hence it is self-induced. The reflex arc is a negative feedback mechanism preventing too much tension on the muscle and tendon. When the muscle tension increases, signals from Golgi tendon organs are transmitted through group of 1B fibers to spinal cord. This signals excites inhibitory interneuron which inhibit alpha motor neuron, so relaxation of muscles occur. Golgi tendon reflex, alpha motor neuron, lobe 1B afferent, extrafusal muscle. The Golgi tendon organ. First, Golgi tendon sense excessive tension. Then sensory neuron produces action potential. Sensory neurons synapse with the interneurons. Alpha motor neurons to quadriceps is inhibited while the alpha motor neurons to the hamstring is stimulated. Then last, the hamstring contracts and the quadriceps relax. Physiological role, protective function from damaging forceful contraction. By damaging forceful contraction, regulation of tension during normal muscle activity, autogenic inhibition, the force generated when the muscle contracts contracts in its stimulus for its own relaxation. Mechanoreceptors are specialized sensory receptors that respond to mechanical stimuli such as pressure, stretch, and vibration. They are located throughout the body and play a critical role in the sense of touch, proprioception, and awareness of one's body position and movement. And other sensory functions. Mesenteric corpuscle, Pacinian corpuscles, Ruffini corpuscles, Merkel cells, Golgi tendon organs, baroreceptors, stretch receptors, giant receptors, muscle spindles, and hair cells. Mesenteric corpuscles are located in the skin, particularly in areas that are sensitive to light touch such as the fingertips, lips, and nipples. They play a crucial role in the sense of touch and tactile discrimination. Structure, here we could see. It's an encapsulated structure, consists of a stack of flattened Schwann cells which surround a group of axons that make up the sensory nerve ending. The entire structure is surrounded by connective tissue and is oriented parallel to the surface of the skin. The sensory nerve fibers are myelinated and are relatively small in diameter. Then stimulation mechanism. Mesenteric corpuscles are stimulated by mechanical forces such as pressure, stretch, and vibration. When a mechanical force is applied to the skin, it deforms the corpuscle and causes the axons within it to be compressed or stretched. This mechanical deformation leads to the opening or closing of ion channels in the sensory nerve ending, which generates an electrical signal known as receptor potential. The magnitude of the receptor potential is proportional to the magnitude of the mechanical stimulus. Function is detection of light, touch, tactile discrimination, recognize objects, perception of shapes, edges and texture, perception of vibration. And Pacinian corpuscles are also like this, located in various parts of body, including the mesenteries and peritoneum. They play a crucial role in sense of touch and detection of pressure. Lamellae, layers of membrane, layers like an onion, then capsule, single nerve fiber, melanin sensory neuron and blood vessel. Structure is kind of similar. Stack of concentric layers of connective tissue and flattened cells known as lamellae. At the center of corpuscle is an axon that makes up a sensory nerve ending. Corpus capsule of connective tissue, lamellae separated by gel, single neuron ending, hyaline sheath, axon of sensory neuron. Stimulation mechanism is similar to the earlier mentioned one. These corpuscles are stimulated by mechanical force such as pressure and vibration. When a mechanical force is applied to the skin, it deforms the corpuscle and causes the lamellae to move relative to each other. This mechanical deformation leads to the opening of closing of ion channels in the sensory nerve ending, which generates the electrical signal known as a receptor potential. The magnitude of the receptor potential is proportional to the magnitude of the mechanical stimulus. Detecting deep pressure, detecting high frequency vibration, detecting the magnitude and the direction of pressure. Proprioception helps to maintain balance and coordination by providing information about joint position and movement, modulation of pain perception, etc. Then Pacinian corpuscles, lamellar cells, Mesinous corpuscles are also there. Ruffini corpuscles. These are like this specialized mechanoreceptors found in skin and mucous membrane. Their structure: bundle of collagen fibers connected to collagen of dermis, dendrites, afferent nerve fiber, Ruffini corpuscles. These are elongated, spindle-shaped structures that consist of network of collagen fibers and a sensory nerve ending, encapsulated in connective tissue and are oriented parallel to the skin surface. Myelinated and relatively small. And stimulation mechanism is similar. Functions: detecting changes in skin and tissue deformations such as stretching and pulling, maintaining grip, controlling forces of movement. Proprioception: provide information about joint position and movement, maintaining balance and coordination. Next one, Merkel cells, located in cells of skin and hair follicles. Structure: epidermis, Meissner's myelinated nerve, dermis, Merkel cells. Located in the dermis, outermost layer of the skin, they are found in the basal layers of epidermis. There are deepest layers of epidermis. Merkel cells are closely associated with sensory nerve fibers. Functions: perception of touch, detection of light touch, formation of tactile sensation, sensory adaptation, development of skin appendages. Merkel cells on Merkel tactile disc respond to touch stimuli. Sensory neuron, Merkel tactile disc. Golgi tendon organ are specialized sensory receptors located within tendons, which are fibrous connective tissue that attach muscles to bones. They are responsible for detecting changes in muscle tension and transmitting this information to the central nervous system, which is important for proprioception and controlling movement. Golgi tendon organs. Golgi tendon organs are encapsulated structures that are located at the junction of tendon and a muscle. They consist of collagen fibers that are wrapped around sensory nerve endings. The collagen fibers are connected to the muscle fibers at one end and to the tendon at the other end. When the muscle contracts, the tension in the tendon increases, which stretches the collagen fibers and activates the sensory nerve endings. Golgi tendon organ. Stimulation mechanism is similar to the earlier mentioned one. Proprioception, control of muscle contraction, feedback during movement, adaptation to changes in muscle tension. Baroreceptors detect changes in blood vessel, blood pressure, and transmit this information to the central nervous system. They are located in the walls of blood vessels, particularly in aorta and carotid sinus. They consist of specialized nerve endings that are sensitive to changes in the structure or tension of the blood vessel walls. Functions: control of blood pressure, regulation of cardiac function, response to orthostatic stress, adaptation to chronic changes in blood pressure. Then stretch receptors, stretching or elongation of tissues such as muscles, tendons, and skin. They play a crucial role in regulating various physiological processes, including movement, balance, and breathing. The sensory organ and the sensory nerve fibers. These are two parts. Stretch receptors. Stimulation mechanism. Stretching of the sensory organ causes the sensory nerve fibers to become depolarized, which generates an electrical signal that travels along the nerve fiber and reaches the brain. The brain then processes the signal and generates an appropriate response, which can include muscle contraction, adjustment of posture, and regulation of breathing. Regulating various physiological processes, including movement, balance, and breathing, provide sensory feedback for brain regulation of muscle tone, prevention of overstretching of muscle during movement, regulation of breathing, like that. Joint receptor, positional movement of joints, which is important for maintaining proper movement and posture. Type 1 and type 2 receptors are there, joint receptors. Type 1 receptors are located in the ligaments and joint capsules and are sensitive to mechanical deformation, while type 2 receptors are located in the cartilage and are sensitive to change in pressure. Type 1 receptors are encapsulated by connective tissue and consist of a central core of sensory fibers surrounded by a capsule of collagen fibers. 2 are encapsulated by connective tissue and consist of a flattened capsule with a central core of sensory fibers. Stimulation mechanism, similar to the earlier one. Provide sensory feedback to the brain about position and movement of joints. Provide information about joint angle, joint position and movement speed. Then joint stability, degree of force applied to the joint, maintaining posture. Muscle spindle, muscle tissue present located with the muscle tissue. Changes in muscle length and rate of change in length, which are important for proprioception and controlling movement. Structure shown here. Functions: proprioception, control of muscle contraction, feedback during movement, adaptation to changes in muscle length. Hair cells are present here. So columnar cells, hair cells are elongated columnar cells, hair-like, also called stereocilia. They are connected to tip links, which are essential for hair cell function. Perception of sound, detection of head movement, contribute to the sense of balance and spatial orientation. Defects in mechanoreceptors, sensory deficits, peripheral neuropathy, motor deficits, damage to muscle spindle lead to muscle weakness, then physiological deficits, damage to baroreceptors. Diseases related to defects in mechanoreceptors. Diseases: peripheral neuropathy, carpal tunnel syndrome, tendonitis, Parkinson's disease.